Recombinant DNA technology is an amazing field of study and research, and it holds a lot of potential for us. If we begin by talking about biotechnology and we introduce that particular term, that is something that really, when we get down to it, has been in place or something that we have used for centuries. It is described as the use of microorganisms to make practical products. And if you look here on this slide, we can see that there are a number of practical products, food products that we do eat or drink on a regular basis that do use microbes to actually produce them. The yeast that we use for bread would be an example of biotechnology. It's certainly used to make various alcoholic beverages like wine and beer. And then you see cheese is another one of those. We could throw buttermilk and yogurt into this mix also. Other ways that we do use microbes to produce practical products is with the production of various chemicals such as acetone, butanol, and then also various antibiotics are made that way. So that's the study of biotechnology. And if we talk about recombinant DNA technology, this is really taking it a step further because this is going to be technology that involves the modification of the genomes, the DNA sequence of organisms for practical purposes. And of course, this provides a lot of potential benefits to us as well. The overall goals of DNA technology is going to be, first of all, to eliminate undesirable phenotypic traits, and that would be in humans, animals, plants, microbes, so in all types of organisms. So get rid of those things that might be detrimental, might cause them to not survive as well, might cause a particular food to not taste as good as it might potentially taste. Another thing is that we can combine beneficial traits from two or more organisms. We can take those beneficial traits and we can combine them into a single organism to hopefully get something that's going to be a little bit better. And then another one is that we can create new organisms that synthesize products, proteins, drugs that humans are actually going to need or benefit from. So those are some of the reasons why recombinant DNA technology is used. Now, this slide right here shows us just an overview of how this might actually take place. We have to first identify a gene of interest in a particular organism. And you can see that right here, we've got, it's shown in green, a gene of interest. This is what we want. And what we're going to do is we're going to take that sequence and we're going to initially cut it out. So you can see that there is going to be a cutting up or a digestion of that DNA. And we get the one that we want, which you can see right here. So this contains the sequence that we desire, and we're going to take that and actually insert it into this bacterial plasmid right here. Again, this is just an example of how this might take place. So we take the plasmid, we put the gene we want inside of that, and then we're going to put that plasmid into a bacterial cell. So on the bottom there, we see that we've got a bacteria that has the plasmid that we desire inside of it. We're going to grow that bacteria, culture it, and then once we've got the bacteria that have that inside of it, hopefully they are going to produce that gene product. And we can then isolate that gene product, purify the protein from it, and we can really use that for all different types of things. So that's the general process of recombinant DNA technology, how it's going to work. If we now talk about some of the tools that would be used for this process, there's a long list of tools, and we'll just talk about some of the kind of most common ones that would be used. First of all, we can use mutagens to do this. Mutagens are going to be physical and chemical agents that produce mutations. And just as a review, a mutation is a change in the DNA or a change in the genetic material. And so if we deliberately change the microbe's genome, so the DNA of these particular microorganisms, we can potentially change the phenotype of those organisms. Now, when we use a mutagen, typically they're very random changes that we're getting. So sometimes it might be something that alters the genome in a negative way, and that's not going to really be very useful to us, but it might also have the potential to change them in a positive way. Okay, so mutagens can be used for that. Another thing that we use is an enzyme called reverse transcriptase. Now this enzyme, notice it has ACE on the end of its name. That is always something that indicates to us that we are looking at an enzyme. And this particular enzyme is something that a lot of different viruses produce, but we use it to our advantage when we're talking about recombinant DNA technology. What this enzyme is capable of doing is it's able to make a complementary DNA molecule, which we call cDNA, and it's able to do that from an RNA template. So if you look at the image here, we're taking RNA and we're converting that into DNA, which tends to be the opposite direction that things are done. If you think about the central dogma of genetics or the central dogma of molecular biology, typically we're going DNA to RNA and we're not going in the other direction. But this enzyme makes it possible. So it is commonly used to create cDNA from isolated mRNA molecules. Another example of a tool that we can use with recombinant DNA technology is that we can actually make synthetic nucleic acids. So these are going to be artificially created DNA and RNA, and we say that they are created in vitro. And that term in vitro means really in an artificial way, so in a test tube environment or in a beaker. That's what in vitro is referring to. So we have equipment that can make these, and there's lots of different uses for these synthetic nucleic acids. So just to give you a few of the uses, first off, they can be used to elucidate the genetic code. So if we are trying to figure out what the genetic code is, we can use nucleic acids for that, synthetic ones for that. It certainly has been used to figure out the genetic code, that big table, the genetic code table. And if we identify new organisms, we can use it for that. We can also create genes for specific proteins that we want to produce. So that can be done. We can create DNA and RNA probes. Those are specific sequences that would stick to, adhere to sequences that we're interested in identifying, so they can help us to locate them, figure out where they actually happen to be within a genome or within an organism. And then we can also use them to create antisense nucleic acid molecules. Antisense nucleic acid molecules are going to be ones that are complementary. They run in the opposite direction of the one that we are interested in, and they tend to stick to those, and they are used many times to negatively regulate gene expression. And what we mean by that is they are used to decrease gene expression or protein production in many cases. Another one, very common one, is the use of restriction enzymes. Restriction enzymes, there's a big group of them really. It's not just one enzyme, but these are going to cut DNA at very specific sites, very specific locations, which we do refer to as restriction sites. Restriction sites are DNA sequences. They tend to be palindromes. And what we mean by palindromes is that if you read it this direction, you will get the same sequence as if you read it in this direction. So both ways, it's reading exactly the same. If we look at an example of one, the letters that you see actually, written in here, they represent a palindromic sequence. Notice that if we read across the top, we get GAA, TTC, and then if we read on the opposite strand in the other direction, we get the exact same thing. So that's the palindrome that we're talking about, and a lot of the restriction enzymes, their sequences are going to be like this, where it's both directions are going to read exactly the same. This enzyme, just to show you how it's going to work, its name is EcoR1. The names are usually according to the organism that they're actually first identified in, and this one was identified in E. coli, so that's where the eco part comes from. And so we take the DNA sequence, we incubate it with this particular enzyme, and the enzyme is going to cut anywhere that it sees that specific DNA sequence. And there might be multiple locations where it actually sees that sequence, but notice on the bottom that it does cut it in a very specific way. And now we've got these two ends that we can work with a little bit further. And what we actually do with those most of the time is we're going to take them and we're going to clone them with some other DNA sequence. Now, enzymes, these restriction enzymes, sometimes they are called restriction endonucleases because they cut inside the DNA, and they fall into two categories. We have ones that we call sticky end restriction enzymes, and then we have blunt end restriction enzymes. And the difference is the way that they cut, the type of ends that they produce. Notice that with the sticky end restriction enzymes, you get what we call an overhang on your DNA molecule. So you end up with something like this, okay? And we can see that in the picture. So that's what the sticky end ones will do. And then the blunt ends, obviously we're producing blunt ends, just like it sounds. So they cut them so that it's the same length on both of the strands. Two types, and both types can be used for cloning purposes. So here we see on the first picture over here that we can use a sticky end enzyme. We can cut two different DNA pieces. So we've got one that's blue or purple over here that we've cut with an enzyme, and then we've got another molecule, it's green, and we've cut it with the same enzyme. And when we cut them, they are going to have ends that want to match up with each other. And so they have been stuck back together down here using that first cutting with the same enzyme, and then we use DNA ligase, and ligase is going to connect the pieces together. So it's really just kind of sealing them together. So first they stick, and then we've got to make those final connections, and DNA ligase is going to do that. You can see on the other side here, we're cutting with a blunt restriction enzyme, and so we can cut the two different ones with the blunt enzymes, and then we stick them together again using ligase enzyme. It's not as easy to clone or stick things together using the blunt restriction enzymes, just because they don't tend to just stick and hold themselves together for the ligase to work, but it certainly can be done. Vectors do need to be used in many cases. Vectors are going to be nucleic acid molecules that will be used to deliver specifically a gene into a cell. So this is going to be a piece of DNA that we're sticking our gene into. These include viral genomes, they include transposons and plasmids, so a lot of different types of vectors, and which vector is chosen depends on really what our overall goal is, maybe the size of the DNA piece that we're trying to insert inside of a cell. All of those will be factors that go into our actual choice. Now, things that are important for vectors, which is going to make them useful, first off, is they need to be small enough that we can manipulate them in a laboratory. So we're not going to take an organism's entire genome and use that as a vector. Typically, we want something that's going to be much smaller. Another thing that's important is that they need to be stable enough to survive inside of a cell. We don't want to put them into a cell and then that cell actually take them apart and digest them. We want them to be able to stay inside the cell, and then we also want them to be able to be replicated by that particular cell. And in addition to replicating, we want them to actually make the gene product. We want them to use the gene for transcription and translation. We need them to contain a recognizable genetic marker as well. This will help us to specifically identify the cells that do have the gene of interest in them. The markers that we use, they are something that in many cases is going to allow the specific survival of cells that contain this gene compared to other ones. It might be something like antibiotic resistance. So if we have an antibiotic resistance gene inside that vector as well, we can take them, we can put the bacterial cells on some media that has the antibiotic in it, and if the cells don't have the vector, they die. If they have the vector, they survive. And then another one is we do want to ensure that there's going to be gene expression of this gene that we put in there. And so by having genetic expression of it, we're saying it is going to be transcribed, it is going to be translated, so you are going to get that ultimate gene product, which is going to be our protein in most cases. Here's a look at the use of a plasmid vector. So plasmids are small circles of DNA. These are things that bacteria regularly produce anyway, but vectors are going to be modified for the purpose of delivering genes. So the orange that we see here, that is the plasmid vector. Notice that it does have an antibiotic resistance gene on it. So this is going to be for us to select the ones that actually have the plasmid. So select the bacterial cells that have what we want. And then we also have within that DNA sequence a specific restriction site for an enzyme to cut it at. So we're going to take the plasmid, we're going to incubate it with the restriction enzyme so that we have sticky ends produced. We could do it with blunt ends, but most of the time it's going to be sticky ends. And so we've produced the sticky ends. Then we're going to take our gene of interest, we're going to cut it with the exact same restriction enzyme. Once we've done that, we can incubate our two pieces together, our plasmid and our gene. And when we incubate them together, we're then going to use our ligase, which is going to seal them together. So after we've done that, we've got this vector, which contains our gene of interest, which has been stuck in there. And we take that, we're going to put it into bacterial cells. So we see that happening. And then we take the bacteria and we plate them, we grow them on some selection media. So this is going to be media that does contain the antibiotic, which means that the only surviving cells on that plate are going to be the ones that actually have this plasmid with our gene of interest inside of it. So that's the general way that we are going to use these vectors. We can also have what we call gene libraries. Gene libraries are libraries of genes, so similar to the way that we have libraries of books. These are going to be collections, big collections of bacterial or phage clones, and each of these clones is going to contain a portion of the genetic material of interest. So if we look at this, for example, if we want to make a library of the genome of this particular bacteria that we see right here, we're going to take that bacterial chromosome and initially it's going to be digested into smaller pieces. So we see the smaller pieces there, the orange. Then we're going to take all those smaller pieces. They are going to be cloned or inserted into a plasmid, and that would be done similar to what we just looked at. And then we will take that collection of plasmids that all have different pieces of the genome and we're going to put them into bacteria. Once we have that, we can basically grow those bacterial cells and we have just this collection of the DNA sequences from that genome that we were starting with. So this is done many times to produce just a collection of fragments from a particular organism, maybe a collection of fragments that are transcribed under specific conditions. So that would be something that we call maybe a Cdna library, because we would be starting with mRNA and turning it into DNA and then cloning it. So it can be used a lot of different ways.